Well, first of all, apologies to all of you who have come in person or are attending online. You will have seen that none of the speakers in this symposium have been able to make it in person, and we won't be able to interact with you online either. So please feel free to email any of us if you have still any questions at the end of our talks. My own presentation is on the effect associated with SARS-CoV-2 infection, which we found were associated with changes in brain structure in the UK Biobank. I'm Gwenaelle Douaud. I'm a part of the FirmUp Center, which is itself part of the bigger Wellcome Center for Integrative Neuroimaging, which is part of the University of Oxford. So I don't know how many of you will have seen already many slides on this subject. There is an increased focus on the effects of mild to moderate COVID-19 in the longer term, and that's been true already when we started working on these paper and these analyses a year ago. But of course, also increased focus on brain-related pathology with neurological and cognitive deficits, for instance, gustatory impairment, hyposmia, impacts shown in radiological and postmortem analysis, and then this heavily debated presence of the coronavirus itself in the central nervous system. But back then, especially a year ago, brain imaging studies had really focused on acute cases, so hospitalized participants or patients who had been hospitalized, and radiological reports of single cases or case series based on CT, PET, and MRI scans. Brain imaging studies had also all scan participants post-infection, so just looked at a snapshot after they had been infected. And that's really where the UK Biobank imaging study can make such a big difference. So for those of you who do not know about the UK Biobank, this is a long-term prospective epidemiological study with information containing genetics, blood samples, lifestyle information, diet, etc., in over 500,000 UK participants, middle-aged and older UK participants. And the imaging data will be available in 100,000 of them. When I started giving talks on the UK Biobank, we had passed our 10,000 participants, and that was probably around 2016. And then only 3 years later, before the pandemic hit, we had passed 40,000 participants. So more than 30,000 had been scanned across the 3 different imaging centers for the UK Biobank in the meantime. Then obviously everything was stopped during the pandemic. So what was the idea here for the UK Biobank COVID-19 imaging study? Well, we had 43,000 participants for whom we had brain scans before the start of this pandemic. And so starting February 2021, hundreds of these participants were invited, so on a volunteering basis, invited back for a second scan. The whole idea here really was to investigate the longitudinal effects of SARS-CoV-2 infection by comparing the imaging scans taken from participants before and after the infection. So compare these changes, if you want, between the control group, so the non-infected participants, and those who have been infected by SARS-CoV-2. So compare the changes between the 2 scans, one taken before the start of the pandemic and one after, during the pandemic and after infection. The whole idea here is really that getting a baseline scan before infection can help us disentangle the contribution of the pathogenic process related to the SARS-CoV-2 infection from pre-existing differences in the brain of participants who can go on to contract COVID-19. So what did we have? We had participants for whom we validated the COVID-19 status either positive based on health records or 2 positive antibody lateral flow test, or negative based on the same criteria. And at the time when we froze the study back in June 2021, we had 401 SARS-CoV-2 participants with 2 usable scans, one before the pandemic, obviously, and one during the pandemic in 2021. But importantly, 96% of the cases had mild infection. In other words, only 15 of these 401 participants, infected participants, had been hospitalized due to COVID-19. And on average, the second scan was acquired on average 4.5 months after infection. This was matched to a control group, which was matched for age, sex, interval between the two scans, ethnicity, and location of the first imaging assessment. As I said, there are 3 imaging assessment locations with the UK Biobank. So we got 384 controls for these. And these controls were matched beyond this demographic data. They were matched for most of the risk factors we could think of: systolic blood pressure, diastolic blood pressure, diabetes, weight, BMI, alcohol intake, tobacco smoking, of course, socioeconomic status. But also, when we looked at the full array of all non-imaging phenotypes, more than 7,000 of them, we found no significant differences. But obviously, correcting for multiple comparisons across so many variables is quite a blunt tool. So please have a look at our paper for more in-depth and more subtle kind of analysis, corollary analysis on this. So what did we found? Well, we adopted 2 different approaches: one hypothesis-driven, focusing on the limbic olfactory-related regions, and one exploratory approach, which covered the entirety of the brain. And both really confirmed the involvement of these olfactory-related regions. In fact, we had 67 significant results for the exploratory approach, and half of these were in these limbic olfactory-related regions. And all of these results indicated greater tissue damage, whether by proxy of diffusion imaging or loss of contrast between gray and white matter, and increased tissue loss, volume or thickness in SARS-CoV-2 group. And that was true, for instance, in the left parahippocampal gyrus, as you can see here on the left, but also particularly if you look at this sliding window that we took 10 years of sliding window, looking at age, when the cases, so the infected participants here in orange, are actually quite similar to the controls in their 50s, this starts really diverging quite considerably in their 70s. And that's the same whichever regions we're looking at. Left orbital frontal cortex, these diffusion indices inside the functional network of the primary olfactory regions, temporal piriform cortex, olfactory, tubercle. Again, something quite similar maybe in their 50s, but these greater tissue damage and tissue atrophy is more marked really at higher ages. That was true not only in the limbic olfactory regions, but also overall when looking at the ratio of the brain to the total intracranial volume, so global shrinking of the volume of the brain, but also in other regions, such as, for instance, the cross-2 of the cerebellum, which is this very cognitive, higher order part of the cerebellum. We looked, for instance, at the cortical thickness reduction, so greater atrophy again in the SARS-CoV-2 group. And we could see in the cortical surface here, over the entire cortical surface, that indeed most of the results are located around these limbic olfactory regions, the parahippocampal gyrus, the orbital frontal cortex, but it also extends in regions such as the anterior cingulate cortex or the anterior insula and the temporal pole, the supramarginal gyrus, etc. And these results really remain significant when we excluded all of the hospitalized participants. So that was absolutely still true when looking only at the mild infection cases. When in turn we compared the hospitalized cases, only 15 of them, remember, compared with the non-hospitalized cases, we found the same pattern again in the same region, so greater atrophy in the hospitalized cases in the parahippocampal gyrus, orbital frontal cortex, anterior cingulate cortex, but it also vastly extended, as you can see here in the frontal, parietal regions and temporal areas, essentially higher order again. We then turn into looking at cognitive differences in the SARS-CoV-2 group. We focus on 10 cognitive scores, and for these we selected these cognitive scores based on how much differences they were showing in an out of sample of healthy controls, or at least non-demented controls, compared with people at risk for dementia or who had dementia. And based on these 10 cognitive scores, what we found was, again, a greater cognitive decline in the SARS-CoV-2 group. So obviously there is a small cognitive decline happening in the controls as well. So 3 years has passed by, but you can see that this greater cognitive decline is quite marked again in participants in their 70s here, for the time to complete the numeric and alphanumeric trials of the trail-making test. And it goes even to showing a difference of a greater, an additional, if you want, time taken to complete that task of 30% for the participants, the infected participants in their 70s. Once more, the results remain the same after excluding the 15 hospitalized cases. So that was absolutely true in the mild infected participants. And in turn, we could relate the duration to complete the trail B with the greater atrophy that we found in a cross-2 of the cerebellum. So what have we found here? Well, we have really something that's quite important, which is the first longitudinal imaging study where SARS-CoV-2 positive participants were scanned before the infection. And that is really important because the pattern of the results is slightly different. If you just look at that snapshot post-infection, it tells you a slightly different story from if you look at the differential between the 2 time points across the 2 groups, of course. And so this longitudinal analysis revealed a significant deleterious impact associated with SARS-CoV-2 on the brain. And this was mainly in the olfactory cortical system. We also found an additional diffuse loss of gray matter on top of these more regional effects. And the hospitalized cases showed greater gray matter loss than the non-hospitalized cases, which is perhaps unsurprising. But I should point out this was a trend. This was not significant because the hospitalized cases, we had only 15 of them. So that was really a power issue here. And finally, we found greater cognitive decline in both processing speed and executive function, really markedly in the trail-making test for the alphanumeric test with SARS-CoV-2 infection. The limitations, the clear limitation of this study are, first of all, lack of clinical severity stratification. We didn't have, apart from being able to distinguish the patients who had been hospitalized from the participants who had not been. There was no other kind of way for us to assess the severity of their clinical symptoms. And indeed, we didn't have the information about whether there were ongoing any symptoms, when they were scanned 4.5 months after their initial infection. So we didn't know if they had, importantly, anosmia, or if they had, signs of long COVID, such as brain fog. We also didn't have the information on the vaccination status of these participants, but I should say that the vast majority of them had been infected before vaccination had become available in January 2021 in the UK. There was only a small number of BAM participants, which is really a bias that pre-exists already in the UK Biobank. And this data only covers the first and second wave, so the original strain of SARS-CoV-2, as well as the alpha, mainly, and beta and gamma variants. And the numbers were too small to be able to contrast the effects of the two, because really the clinical presentation tends to be a bit different between these different variants, but also now we know as well with Omicron. And finally, the MRI acquisition was the one developed for UK Biobank, so had not been optimized for the olfactory bulbs in the primary olfactory cortex. So the million-dollar question is really, why is it in the olfactory cortex? Well, some people have obviously proposed that there was neurotropism, so invasion of the virus in the brain, but there's obviously conflicting evidence at the moment, possibly depending on the degree of severity or the kind of variants. Maybe a lot of things will be resolved in the coming months or years. Could also be an anterograde degeneration starting with the olfactory neurons, so no need to actually for the virus to get into the brain itself. More and more evidence point out really neuroinflammation, the kind of effects that we see also, for instance, with influenza. And of course, it could be also at least some of these effects seen in the brain around the olfactory regions could be related to sensory deprivation due to hyposmia or anosmia or loss of gustatory sense. That really in turn brings the possibility that if this is due to, or at least some of it is due to sensory deprivation, when these senses are recovered, hopefully over time, after a few weeks, after a few months, then it might be that these differences in the brain, these abnormalities in the brain are reversible. And there are for these some small studies that point out some evidence that there is some normalization happening in the brain after 6 months. Doesn't completely resolve. Obviously, there's still some residual differences, but perhaps some normalization in a few of the patients that have been followed for that long with PET scanning. And on that cheery note, I would like to thank all of my collaborators in Oxford, in London, at the NIH, especially Sujin Lee and Steve Smith, who have put an insane amount of work into that paper and that study. Our founders, the Wellcome Trust, and thank you very much for your attention. Please feel free to send me an email if you have any questions, because unfortunately we cannot interact either in person or online here with this symposium. Thank you very much.